Hello everyone. So this is our fourth lecture as a part of our revision series for the upcoming Prelims 2025, and the theme chosen for today's discussion is from Science and Tech, a very specific area about the science and tech that we find around us in our daily life. Right? Many of the aspirants, when you read current affairs compilations, you start reading about high-tech concepts, you start reading about quantum computing, biotechnology, without realizing that the question asked from all these areas would be very superficial in the prelims examination. But it's the questions that are asked from science that you encounter in your daily lives around you, where the questions are slightly deeper. Right? When you sit in your car and when you cross toll booths, have you ever thought how RFID, the radio tag, that fast tag that is there on your car, how does it work? What is the mechanism through which it works? Have you ever given a thought to that? UPSC would actually explore that deeper. And when you're down with fever, when you take a course of antibiotics, have you ever thought why does the antibiotic kill the bacteria but it does not kill the human cells? Have you ever thought on those lines? And when you travel by an aircraft, have you ever wondered about the mechanism through which the lift is achieved by the aircraft? Why does the aircraft have to accelerate to a certain distance before the lift can be achieved? Right? So these are the thoughts. When they are triggered, you should have had the patience, you should have had the curiosity to just spend 5 minutes browsing about everything that you encounter in your daily life. That is all that it takes to prepare for science and tech. UPSC doesn't really want only IITians or people great with science. If that was the case, UPSC can simply go to campus recruitment to IITs and it can just induct officers from there. But UPSC wants people from sociology background, from history background. And UPSC also wants engineers and science students from tier 2, tier 3 colleges as well. And that's the reason why UPSC makes science questions very general in nature. And trust me, every topper who cracks his exam, who cracks prelims beyond with a score of 100 plus marks in prelims, they would have all aced in science and tech questions. And they are not only limited to science students, even students from economics, sociology can also get 100 plus by getting some 70% or 80% questions in science and tech right, and especially questions like what was asked last year in 2024 prelims. It actually made science and tech questions easier for most aspirants. Right? So it's a very fertile area to capitalize on, and you should be doing it. So we start our discussion on science and tech, starting from this particular theme on science and tech involved in our daily life around us. Right? So that can be easy to relate to, instead of directly starting our session first from quantum computing or from space technologies. Instead, we start with simple ideas as how mobile phone wireless charging works. Right? We start with simple ideas as to how oxygen levels are measured in oximeters. Right? Nowadays, all of us, we have wearable devices, we wear smartwatches in which the heart rate is continuously monitored. Have you thought, why does this green light blink? Is a green light or a red light which is better to measure heart rates? Right? There is just so much of tech which is very intrusive in our daily lives, which we are very dismissive about, we are very casual about it, we just are lazy to explore it a level deeper. Right? So that's going to be the objective of today's session. The reason for that is because of previous year's questions. Right? And you see, there are questions asked every year. This is a 2023 question. Okay, I think this is 2021 or 22 question. Right? So every year there are at least 1 or 2 questions asked from the daily science around you. Okay, and getting these 1 or 2 questions easy is correct is very easy. So that is the reason why we focus on this. And moreover, science and tech is a subject where you really can ace the examination science questions without even reading a single NCERT. I would actually ask you to even stop reading all current affairs compilations, because when you read compilations, they go to depths unnecessarily on technologies that are not commonplace in our daily life. For example, how many of us have ever seen the applications of biotechnology in our daily life? Right? And how many of us actually require the usage of quantum computing in our daily lives? Maybe for weather forecast you may require it, maybe for large language models, for generative AI you may need supercomputers. Right? But as a common man in our daily life, we don't really need all those technologies. But getting into the depth is what is actually happening in all the current affairs compilations. So basically what happens is you overload yourself, and in the process you become ineffective in differentiating between usable knowledge for this exam and non-usable knowledge. Right? If an IITian were to read all that, if he is a computer engineer and if he reads a paper on some neural networks, he can grasp it, right? And if he reads current affairs compilations on extremely difficult concepts like quantum computing, he can grasp it, right? But you don't have to, if you're a non-computer science student, if you're not really interested into all those frontier technologies, don't be bothered, don't be worried about it. You have questions like how food is cooked in the kitchen, right? And such kind of questions will ensure that your questions, your possibility of getting certain decent number of questions in science and tech correct is also made possible, right? That's why work on what is naturally easier, ignore areas which are actually difficult to master. And moreover, when you look at all these questions, these are all questions on frontier tech, right? For example, this is a question on metaverse. I don't even think anybody who's watching this video would have ever owned a cryptocurrency, or have you ever owned a crypto wallet, or have you ever purchased a property on metaverse, correct? All these are not going to happen, right? So that is why when UPSC asks questions from those areas, it only tests, have you ever in the last 1 or 2 years, has this word metaverse fallen in your ears or not? It doesn't even go into the depth of all that technology and everything. Similarly, I expect that a question this year or in the upcoming year on generative AI or large language models will all be very, very surface level, right? But if at all a question is asked about how your mobile phone cameras work, there could be a certain level of depth to that. Okay, they could ask questions as to what kind of lenses are used in mobile cameras. Is it concave, is it convex, or is it a series of concave and convex? Are prisms used in DSLR camera lenses, right? To that extent UPSC can get into, because it's a commonplace technology. We all have cameras in our pockets, but did we think about how image generation actually happens? Correct? We talk about optical zoom. How is that zoom achieved? Have you ever given a thought about that? Those are the areas which UPSC will focus, right? So that is the reason why the theme for today's session, it's simply going to be the basic science and tech in our daily life. All right. So once this discussion is over, you will be able to get the answers for these questions, right? And some of you may even get these questions right even right now. So what you can do is you can simply pause the video. These are questions which I have framed based on this particular theme. So you can simply pause the video, think about answer for all these questions, right? And this is exactly what we'll be discussing in the next 40 or 50 minutes of our discussion. All right. So I hope you have gone through the questions, and now let's move on and let's start discussing the first theme for today's session. So the first topic that we'll be discussing today would be about wearable tech. So all of us, we have smartwatches, and smartwatches have become incredibly powerful in the recent years. Apple watches, these latest models, I think they can even warn the occurrence of heart attack. Correct? So they have become very powerful. So that's why you need to really understand how smartwatches make certain measurements. They have some sensors with which they are able to measure the oxygen levels of your blood. They have some sensors with which they can measure your heart rate as well. So it's time we try to understand something about how the mechanism actually works. So first, let's take the case of measurement of oxygen level in your blood. You remember during the days of COVID-19, all of us, we had pulse oximeters in our homes, right? And often we used to check what is the saturation levels of oxygen in our blood, right? So how does it actually work? Before we get into that, you need to understand how do our eyes perceive color, right? Why do we perceive apple to be reddish in color? Why do we perceive leaves to be greenish in color? The reason is simple, because white light has various components to it, right? When you send white light through a prism, you can split it into its component different colors. All colors combined together, it becomes white light, right? You have violet, indigo, blue, green, yellow, orange and red. Each color actually corresponds to a particular wavelength of electromagnetic radiation. Red light has a certain wavelength, green light has a certain wavelength, okay? And it is the characteristic of every material to absorb certain wavelength, reflect certain wavelength. So when it comes to apple, what apple does is, it absorbs all other wavelengths. Violet is absorbed, orange is absorbed, blue is absorbed, green is absorbed, only red is reflected back. So that's when the red wavelength, when it actually comes and hits your eyes, we are able to perceive the color of apple to be red. Same is the case with leaves. When it comes to green material, all material which appear to be green in color, what they do is they absorb all other colors into them. Only the green color wavelength alone is reflected back. It reaches our eyes, we perceive green color, right? So this is the basic principle, basic physics based on which the measurement of oxygen in your blood actually happens, right? Now take the case of blood which is rich with oxygen, which we call as the oxygenated blood, right? The hemoglobin actually holds more of oxygen in it, and so the blood appears to be bright red in color. It's like reddish, deep reddish in color, which means that more of red wavelength is reflected back. Okay, you should not visualize it in the reverse. You should not say that a lot of red has been taken in by the body, that is why it's appearing to be red. No, that is not how physics works. That's not how light works. If an object is more reddish, it means that more of red wavelength is given out. It falls into your eyes. It is actually transmitted to your eyes. That's why you're able to see that, right? So oxygenated blood which is bright red means a lot of red color wavelength has been reflected back. It was not absorbed. But you take blood which is actually less oxygenated in content. If there is less amount of oxygen in the blood won't be that bright red, which means that a lot of red wavelength now is absorbed by the blood. So oxygenated blood reflects more red light. Deoxygenated blood absorbs more red light. That's it. So that is the idea behind how we measure oxygen levels. So what do? We send red light from a red light transmitter through a translucent part of our body, right? Our fingertips are translucent. I'm sure you would have observed it as a child. We would have experimented playing with red color. We put red light on one end of the finger, on the other end it's very clearly visible, right? Whereas you take it and put it on your skull on one side, you cannot see the red color on the other side, right? So our fingertips are translucent, our toe tips translucent, ear lobes are translucent. Okay, in all these parts of the body the measurement can be done. So what do? We send the red light from the top and it passes through our blood vessels, and when it passes through our blood vessels, if the blood is nicely oxygenated, right, in that case more amount of red light will be reflected back. It will not be absorbed by the body. That's the principle. So on the other end what we have is a receiver, a sensor. Okay, so if we have sent certain amounts of red light and the same amount of red light has been received by the sensor, it means that the blood is 100% oxygenated, right? There has not been any absorption at all. Whatever light was transmitted, it was all reflected back. So the blood is well oxygenated, correct? Whereas if the amount of light that was sent into the blood vessel and the amount of light that was collected by the sensor, if there is a difference, it means that the blood may be slightly less oxygenated. Okay, and depending upon the variations, we will be able to make measurements as to how much of oxygenation is present in the blood, right? So instead of simply using only red light, we also use infrared light also. Okay, infrared light works in the exact opposite way. If it is nicely oxygenated, it means that more amount of infrared will be absorbed by the body. Okay, if it is a deoxygenated blood, more amount of infrared will be reflected by the body. Okay, so this is the logic. So we do twin corroborations. We not only use only the red light, we also apply infrared also, so as to get double confirmation that this is the extent of oxygenation which is present in the blood. Okay, so the measurement of oxygen in pulse oximeters, okay, it actually works on the principle of amount of red light that is absorbed or reflected by oxygenated and deoxygenated blood respectively. Okay, I hope it is clear. Now we move on to how is heart rate measured using our smartwatches. What is the logic behind that? Now coming to the measurement of heartbeat, what our smartwatches do is basically they depend upon the principle of absorption of green light. Okay, so you take your smartwatch and it's nicely located in the appropriate place for measuring heartbeat, which is your wrist, right? The reason why doctors for ages, why have they measured pulse from your wrist? It's because it is at the wrist where the artery and the veins are closest to the surface, right? You take the case of your muscle and your biceps, you make a cut to your muscle, you're not going to die because the arteries are deep inside. You'll only simply be making an incision to your skin and your muscles. right? But whereas when it comes to wrist, the arteries and the veins are so close to the surface that even an incision can actually lead to bleeding and death, right? So the arteries are the closest, and it's exactly the place where wrist watches are worn. So the site is appropriate. Now what we do is we use the principle of absorption of green light, and to understand what is this principle of absorption of green light, let me show you an experiment. You take a green light, you focus it on a light receiver. Your mobile phones are very good light receivers. You have apps which can measure the intensity of the light that is received by the front camera, right? So you use such apps. What you do is you transmit green light directly. If there is no medium here, now the light intensity can be measured by the mobile phone. Now what you do is you take a beaker, and in that you pour red solution. It could be wine, it could be beetroot juice, cranberry juice, anything. You just pour it, keep it here. You can find that the intensity of the light that is received by the front camera will actually fall down. You add one more container here, again the intensity drops drastically. The reason is more of red medium starts absorbing the green light, only allowing very less green light to emerge out. Okay, this is the principle that we are going to apply so as to measure the rate at which your heart pumps blood. Imagine whenever your heart squeezes, whenever it contracts, it means immediately blood is sent to the blood vessels. So the blood vessels become dilated, they become bigger, right? And when the heart relaxes, right, it means that the blood vessels also relax, right? Whenever heart pumps, blood is squeezed through the blood vessels, as a result of which it expands. When the heart relaxes, immediately blood vessels contract. Okay, so what you have to imagine is whenever the heart pumps, two beakers are brought in place because more blood. When the heart relaxes, there is just one beaker. Correct? This is what happens to your artery every time your heart pumps. When a heart pumps, immediately the volume of your blood vessel increases, and when your heart relaxes, the volume of your blood vessel relaxes. Okay, so it means that whenever the volume of blood vessel increases, more blood is inside there. So it means that the green light, more of green light will be absorbed, and only less amount of green light even emerges out on the other side. Whereas when the heart relaxes, there is less amount of blood, which means that more green light can come out unabsorbed, right? And on the other side you have a sensor. Okay, so you on one end, if you take the back side of your smartwatch, you have two sensors. One is for the purpose of emitting light. Okay, it's not a sensor, it's just a LED, green LED bulb. It emits green light. Just next to that you have a receiver. Okay, you have a sensor which will receive the amount of green light which comes out unabsorbed. Okay, and this is how you can make a plot. You can make a plot and measure that every time. So you can actually measure how much amount of such cycles of increase in absorption and reduction in absorption have taken place, right? So if 80 different cycles have taken place, it means that 80 times the blood vessel has increased and it has decreased, correct? It means the heart has pumped 80 times per minute. If the cycles of how much green light received by the receiver, you measure the intensity. If the intensity is peaking and it's coming down, if such cycles of peaks and lows are 120, it means that in 120 times your blood vessels have increased and it have contracted, right? So that indicates the rate at which your heart beats. That's all. It's so simple. Okay, so the measurement of heartbeat is based upon the principle of absorption of green light. So when you send green light through a red medium, the green light, majority portion of the green light, it's absorbed, so the intensity of the green light coming out is reduced. So when you have more quantum of red medium, very less light comes out, correct? So when heart pumps, more blood enters into the blood vessel, so more absorption of green light will happen at that moment. Okay, so if more amount of green light is absorbed, very less green light even comes out. So when you mark it on a graph, it is the low point, right? And when the heart relaxes, less blood, so automatically the amount of green light absorbed is also less, so more green light can come out of the blood vessel. So that is measured now, which goes to a peak, right? And then again the cycle, once again it drops, goes to the peak. So how much ever cycles we observe indicates at the rate at which your heart pumps. Alright, so we move on to the next topic. So the next theme that we will be looking into would be how our mobile phone technology works, and a very common technology that's used in mobile phone today is accelerometers, right? So there was also a question asked in 2023 about accelerometers, but I think even though a question had been asked, not many would be very clear about how this technology actually functions, and let me explain it to you how accelerometers work, right? Before we get into how accelerometers work, you need to understand a basic physics concept called as capacitance, right? So this is something that we have studied in our 11th and 12th standard for board exams, right? Basically, when you have two plates and there is a potential difference between the two plates, okay, just consider there are two plates and you connect it to a battery, okay, and there is a certain distance between these two plates, right? And on one plate connected to positive electrode, other plate is connected to the negative terminal, as a result of which there are positive charge which has gone onto one plate and a negative charge accumulation has gone into on the surface of the other plate, right? Positive charges are all deposited here, negative charges are all deposited there, right? So now what happens when you bring the plates closer to each other? As you bring the plates closer and closer to each other, it means that the capacity of the plates to hold charge will increase. The ability of the plates to hold charge increases as the distance between them decreases. There is an inverse relationship between the distance and the ability of the plates to hold charge, right? So this is the idea called as capacitance, which is actually used in your mobile phone in accelerometers. And it is because of these accelerometers today, when you turn your phone, immediately from portrait the screen changes to landscape. It is because of accelerometers today you are able to play your mobile phone games, right? And it is because of accelerometers today you ensure that when you shoot a video, stabilization can be achieved. So all these functions are directly as the result of this physical property, electrical property of capacitance, right? So I hope you are able to understand. Two plates, they hold charges, and these capacitors are actually used in your fans, in motors. Why? They hold enormous amount of charge on the plates, and whenever it is required, suddenly they discharge the entire charge. And when they discharge all the charge at a instant, in an instantaneous moment, such a huge amount of charge can be used to operate appliances which requires initial starting torque. For example, you take the case of your electric fan. Why does it have a capacitor? Simply because to start the fan to start rotating, you need a high discharge of charges, right? That's why whenever the capacitor becomes weak in your fan, the best solution is to go and turn the fan with your finger, right? Once it starts rotating, after that the electric current can sustain the movement, but for giving that initial push you need a sudden discharge, and that is why you have capacitors, because capacitors hold charges, right? So it is from that we understand this idea also called as capacitance, which means that it's the capacity or the ability of the electrodes of the plates to contain charges. So as the distance comes closer and closer, more charges can be held by the plates. This is the idea, right? Now understand that we have a sensor like this which is actually placed in mobile phones, right? So this is a sensor which replicates the action of this capacitor. Okay, so how does it work? Let me just explain it to you. You see, this is a base plate. Okay, so this is just a normal, you can say it's a polymer plate, it's a plastic plate. Okay, on this plastic plate you have these blue color plates. You can consider these are the positive charged plates. All the blue color ones, they are all fixed to the plastic plate. Visualize, right? They have a profile like this and they are just fixed, attached, glued to the plastic base, right? Now you have a rubber band here, you have another rubber band here, and to the rubber band you are fixing another conductor, which is actually the other plate that is used in capacitor. That is what this orange color plate is, right? So basically the orange plate is not fixed. It's not fixed to the plastic board, but rather it is just suspended on two ends using these two rubber bands. So it means that this orange conductor can move up and down, it can move to and fro, right? Whereas the blue color plates are just fixed, they are not movable, they are immobile. Okay, now you are giving positive charge to one plate, you are giving negative charge to the other plate. Basically it's similar to capacitor, right? Now what happens is, let's say I hold this plate and I move in this axis, right? I just hold it and I just walk a few steps, and when I do that, now what happens is, this orange plate, it continues to move because it is freely suspended. I may have come to a halt, but this orange plate will still continue to move because Newton's first law, the body continues to be in a state of rest or in motion, right? So basically, since I have started the motion, the body continues to be in motion, and the rubber band which is there ensures that still the inertia maintains. Okay, so basically what happens is, the distance between the two plates actually reduces, isn't it? This orange plate and the blue plate here, as it comes closer and closer to this side, this distance reduces, and when the distance reduces, it means the capacitance increases. That will be measured by a processing unit. A sensor will measure, okay, earlier capacitance was so much farad, now it has increased to this much amount of farads, right? Farad is basically the unit, right? So this is how changes in capacitance will indicate how much of movement has actually happened on one axis. Understand that this is a linear movement in an axis, the straight line movement in one axis, x axis, right? So imagine if you are playing a game like this. There is a car racing game that you are playing. Earlier, 15-20 years back, when you used to play on computer, even now when you play computer games, the same car race, you use the right and left button, right, to impart the movement to the cars. But when you play the game with the mobile phone, you don't use buttons. Instead, you just simply tilt the mobile phone, right? So imagine when you have a one axis accelerometer like this, and you have a car, you want to move to the right. Now what do? You hold the mobile phone and you walk towards the right. It's only when you move towards the right, because of Newton's first law, this plate would continue to move to this x axis. Correct? But do we actually do that? We don't walk to the right, we don't walk to the left every time. We simply tilt the phone. When you tilt, what is actually happening? You are moving not on an axis, but rather you are moving on a plane. So this is the plane in which your mobile phone is, right? You are watching a mobile phone in the XY plane. Now when you tilt it, you are tilting it again in the XY plane, right? With reference to x axis, you are moving by certain degree. With reference to y axis, you are moving it by certain degree, right? So you are imparting a planar movement. This is not an axial movement. So it means that just having one axis accelerometer is not going to serve your needs. You have to create another accelerometer here, which is placed vertical, which is placed perpendicular to this, so that the movement in the y axis can also be registered by that accelerometer. Correct? So that is what happens when you tilt the phone. There is a movement of the accelerometers in both with reference to x axis as well as y axis. So both the axis, the accelerometers registers some movement. The principle of capacitance will be processed, as a result of which the software will align the image of this car from here to here, or left towards the left, right? So we can even test it. For example, so this is a screen, right? Now what should we do so as to tilt, so as to change the orientation of this image from portrait to landscape? We basically tilt the phone. Correct? So when you tilt the phone, what happens is immediately the orientation changes. So basically you are infusing a movement on the XY plane, and you have two axis accelerometers working inside the phone. But not only that, imagine this image, right? I am revolving the phone, which means that I am imparting a movement in the third axis. If I simply move right and left, this is just a tilt. But imagine now what's happening. I am just tilting the phone, and you see how the image orientation changes. It means that there is a third accelerometer as well, which is operating in the z axis, right? Even the movement in the z axis, basically this overturning movement, it's actually happening in this z axis, which penetrates through me and through the board, right? So it is in this axis, axis we are imparting this movement, whereas when you are imparting this tilting movement to the right and left, we are simply moving the phone on the XY plane. On the x and y axis alone we are moving it. But when you are revolving the phone like this, and still the image orientation changes, it means that this phone has a 3-axis accelerometer, and that's what is the case with normally with all the phones today, right? All the phones, they work on the principle of a 3-axis accelerometer, so that your pitch, your roll, all the movements in the 3-axis is all registered by movement of these plates, right? If not for 3-axis accelerometers, see, for the game like this, for a car racing game on a straight road, you only need to tilt right and left, right? You need to simply move right and left. But you take the case of games like flight simulators. In flight simulators, you are actually flying in the air, right? You need both movement right, left, as well as tumbling movement, as well as upward movement, right? You need movement in all the 3 axes, correct? So to play a game like this, you actually need a 3-axis accelerometer, and that is what is actually there in all the mobile phones today, right? So I hope you are able to understand how accelerometer works. The accelerometer works on the principle of capacitance. Capacitance is basically the ability of a electrically charged plate to hold charges, and the capacitance is basically a function of distance, right? It is inversely related to the distance. That is, when the distance reduces, capacitance increases. When the distance goes higher and higher, capacitance reduces, right? It is one of the factor. The other factor is the voltage applied. The other factor is the medium that is there between the 2 plates. Those are all other factors, but I am saying one of the factors that determines capacitance is the distance. So any changes to the distance influences the capacity of the plates to hold charges, and that is what we are applying here. We are using 2 charged plates, one fixed to the plastic base, other which is suspended between an elastic band. The plate that is suspended on an elastic band can move to and fro on an axis, and this is what is leading to a registry made on how much of capacitance has increased, has it, or decreased, right? So when you have 2 axis accelerometers, you are able to get tilting movements on a plane. When you have the 3rd axis accelerometer, you are able to have effect on all 3 axes, which is what is necessary for playing games like flight simulators, which work in a 3-dimensional space, whereas this car is only working on a 2-dimensional space, right? So I hope this concept is clear. So accelerometer is one technology which has been around, which has been in use even before mobile phones were discovered, right? So very nicely the technology has been embedded in mobile phones. I think Apple was the first company to do that by bringing accelerometers in iPhones, as a result of which we are able to have the first effect where you can shake the phone and have dice which can be rolled, right? All those effects were created in the first iPhone that was brought in because of Apple, right? So I hope this is clear. So we'll move on to and there are also many other technologies which are today present in mobile phones. I would give you assignments just to go and find out what those other 2 technologies is. One is, in addition to accelerometers, mobile phones also use gyroscope, right? So try to find out what is a gyroscope. Gyroscope. Okay. Gyroscope is used in mobile phones. It's used for stabilization purposes, right? It can also do the same work as an accelerometer, just that the motions will be even more smoother. Okay, how is it achieved? Try to find it out. And also there is another instrument which is used in mobile phones, another sensor which is called as magnetometer, right? This doesn't actually use a physical magnet or anything, but find out how magnetometer works. It's only because of magnetometer you are able to find north and south in your phones, but there is no magnet in your phone. But a magnetometer uses a certain kind of a sensor. So that can be taken as an assignment where you can try finding out how magnetometer and a gyroscope functions. And gyroscope has utility in many other applications, in terms of stabilizing your spacecraft, in terms of stabilizing automobiles, right? A lot of functions can be attributed to gyroscope. All right. So we move on to the next topic. Next topic would be about the different payment systems. This is something that we all use in our daily life. We use the debit and credit cards to transact, right? So have you ever thought how does it work, right? And you can make 2 types of transaction with the cards. One is where you can swipe it physically, other one is you don't have to swipe, you have to simply just take it near point of sales machine and automatically it transacts, right? You just have to touch it, tap it, right? So how do both these systems work? Let's have a discussion on that. First is the system where you work with the magnetic stripe, where you swipe your card, right? How does that system work? Before that, if anybody is interested to look and think about this question, here is a question for you, right? Maybe you can pause this video, look into this question, find out what is the answer, right? So how does this magnetic stripe card actually work? How does your debit and credit card, how does it work? Basically, it's a card which has a plastic card. On the plastic card you have a magnetic tape which is pasted, right? This magnetic tape has it has 3 tracks. So that's what you can find here. It has 3 tracks and it has been magnetized in a particular way. Okay, there is a particular way in which the tracks magnetic fields are all kept aligned here. Okay. Now the physics that is actually associated with the working of this system is what is basically Faraday's law. What is Faraday's law? When a magnetic flux is created near a coil, electricity is generated. This is 11th standard knowledge that we have studied, right? What is that we have studied? You take a coil, a copper coil of wire, right? This is called as a solenoid, right? And in this copper solenoid, when you bring a big magnet, you put the magnet inside and you create a to and fro motion. Basically, you are varying the magnetic field. In a constantly varying magnetic field, right, there is a current that is induced in this particular coil. Okay, so that current is now taken to a processing unit. So understand, each tape, right, each tape on a card is magnetized in a particular manner. So each card, when it is swiped near the solenoid, it creates a very unique magnetic field. It creates a very unique electric field, correct? Because it is as per the magnetic alignment which is there in these 3 tracks. Based on that is how the electric signals will be generated, correct? So no 2 cards will have similar magnetic track alignment. So no 2 cards is going to produce similar electric signals. So unique signature electric signals are going to be produced, correct? So basically it is the Faraday's law which is actually being employed in the working of this swipe cards. Basically you have a magnetic tape full of tracks, 3 tracks are there in the magnetic tape, and this card is swiped in a card reader. And what is there inside this card reader? You have a solenoid, and this solenoid, by working on the principle of Faraday's law, as soon as a magnetic field is it's varied near a solenoid, immediately an electric current passes through the solenoid. It is taken to a processor, right? And from this processor, authentication next take place. Okay, so this is one way of how the card payments actually work. It works on the basis of physical contact, right? But then there is a second way of how the payment system works. It works in a non-contact basis. It works on a contactless basis, right? But the technology involved in this is a different technology which is called as RFID, radio frequency identification. That's the same technology that you find used in your FASTag tags that you have in your cars pasted, right? When you cross a toll booth, automatically a transaction happens. How? Because there is a card reader, and the reader reads that radio frequency ID. Okay, as a mechanism actually takes place, which I will be discussing right now. And it's the same RFID tags which you will find in textile showrooms, where each shirt or a t-shirt or a pant, everything has a tag attached to that. Why? Otherwise people will just steal that textile, right? You try stealing a textile from a showroom, as the minute you try exiting the shop, near the entrance of the shop you have 2 RFID readers, right? It will immediately sense that there is a textile which is being attempted to be stolen, and it immediately raises an alarm, correct? So that's why when you go to the billing counter, they remove the tag and then they give it to you. Basically they're removing the RFID tag. So it's the same mechanism which is at work in the toll booth, which is at work in the showrooms, as well as in your credit cards that you carry. What is this RFID mechanism? Let's have a look at that. The next system actually works on the principle of radio frequency identification. What you need as a component for this, just 2 components. On one hand you have what is called as an RFID reader, right? And on the other hand you have what is called as a tag. This tag could be what is placed on your car's windshield, that fast tag, or this tag could be there in your debit card. Right, in some of the debit cards you would have seen that Wi-Fi symbol will be there, which means that it's a contactless card. Right, those cards would be having a RFID tag inside them. Okay, that's what we call, the cards have certain chip in them. Right, it has an integrated circuit. There's a circuit inside that. Right, so this is how a tag looks like. Inside your fast tag, on the fast tag you have some barcode, right, but that barcode is simply for our identification purpose. Okay, or the barcode can be read separately with a barcode scanner, correct, but this barcode has nothing to do with radio frequency identification. When you open the tag, you have a circuitry inside. Right, what is that circuit? Basically you have a copper coil, right, and in addition to that you have what is called as a rectifier. Basically a rectifier converts AC into DC, so that this DC current can be fed to a circuit. Right, in the circuit you have certain memory. Basically that's all. These are the components that you actually find inside a tag. Right, you have what you can see here as a coil nicely pasted, right, and that coil is connected to a rectifier. Basically, because what happens is the output that comes out of a coil is an alternating current. You cannot directly connect alternating current to a chip, right, so we have to make it as a DC first. It is to achieve that we have a rectifier there inside, a rectifier which is actually connected to a memory basically. Right, now basically for this chip, for memory to be accessed, you need electricity. Where is the current going to come from? You don't have any power source in your car's fast tag, you don't have any power source in your ATM cards, right, so that's why these tags are all called as passive tags. Okay, so let's not get into what is active tag and all of that. Mostly the tags that we use in our daily lives, they're all passive tags because they don't have an independent power source. Right, then how are they going to be powered? Okay, so it works on the principle of induction, radio wave induction. Right, what is this? First of all, you should know what is a radio wave. Right, radio wave is a part of the electromagnetic spectrum. It is in the same spectrum you also have microwaves, you have X-rays, you have gamma rays. Right, all of these are what are called as the electromagnetic spectrum. They occupy different positions in the broad spectrum. Right, so basically you should understand these are all carriers of energy. Right, so when you tune in and listen to a radio station, basically the signal that comes to you is basically a form of energy. Right, and how is a radio wave created? We can create it in physics lab. Right, in college days and school days you would have created radio waves by using what is called as oscillators. Right, you connect an oscillator to an antenna, and in the oscillator you can give various signals. You can give pulses, you can give sine waves. Right, by continuously oscillating the current, you will be able to, basically when you supply alternating current to a loose end of wire, which is a loose end of wire is what is called as an antenna. When you supply current, right, radio waves are generated, and those radio waves are now they start transmitting to different directions. Correct. So this reader, the reader that is actually going to read this tag, basically that is going to generate radio waves. That is going to generate radio wave, and that radio wave is going to come. It is going to fall on this antenna. As soon as it touches this antenna, immediately alternating current is produced in the antenna, and that goes to the rectifier, becomes a DC, goes to the memory, retrieves the memory. Right. So from here, the wave, the radio signal is sent back to the reader, carrying the memory which is there in the tag. That memory, when it goes to this reader, now that memory will be matched, right, and it will be seen if the memory fits into the database which we already have. So if your fast tag has a balance of 1000 rupees, that is actually there in that memory. Correct. So that memory will now go to the reader and it will be authenticated. Right, so it is simple. It's basically working on the principle of radio wave generation and radio wave propagation. Right, so radio wave is created in a RFID reader. Now that goes and touches this antenna, powers the circuit, and from there a return signal is sent, and that return signal goes and it is collected in this RFID reader. Now that is matched with the database. Simple as that, right? So the only idea that you should know here when it comes to working of this RFID mechanism is that this is a passive tag. There is no independent power source. Then how does it get energized? It's because of the incoming electromagnetic wave, that is the radio wave, which is actually an energy wave that carries an energy, right? So basically when you use, in the olden days when you used to listen to radio and all of that, you start tuning the radio, correct? You can actually hear something, but what happens is that alone is not loud enough. That is why we have independent power source in our home. We use battery or we plug into the socket, and what is the purpose of all of that? It is just going to amplify the signal that is coming through the radio wave, right? So radio wave itself is a form of a power source. That itself, mild electric signals can be got, but just that is not loud enough to make the speaker oscillate and work. So that is why we do external power supply to it, so that we amplify the signal and make it loud enough for everybody to hear, right? So this is the concept behind how contactless payment systems work, right? And as an addition to that, we also have the wireless charging technology that we find used, right? We just take our mobile phones, we keep it on a plate and it starts getting charged. Okay, we have wireless systems of charging in different cars which are coming in the recent days as well, right? So it works on the principle of what is called as magnetic induction. Okay, this is the principle that I was actually telling you as when I was explaining to you how these magnetic card readers, how do they work, right? This is the solenoid and you have a magnet inside. When you move the magnet, automatically current is generated, correct? Now consider you have a battery and this battery is producing direct current. Okay, with direct current you cannot create a magnetic field. So what you do is you supply this direct current to a inverter. Okay, in our homes we have inverters. What is the purpose of inverter? It's a power backup supply when power goes off, right? So we have batteries. Batteries are connected to the inverter. Battery gives a DC. Now it goes to the inverter, it becomes alternating current, and this AC comes to light, fan and everything, and that's how appliances work, right? Similarly, there is an inverter circuit here. This inverter makes this DC into AC, and this alternating current is now what goes into this coil. When this alternating current goes into this coil, right, now because alternating current, the direction keeps on changing, so it means that the magnetic flux that is created in the coil will also keep on changing. Okay, it's because of varying nature of the alternating current, a magnetic flux is now created. Okay, now the reverse process happens, what I have told you here. When a magnetic field is created and this magnetic field is brought near this coil, electricity is generated, correct? This is what is happening in this particular coil. In this particular coil, the magnetic field created in the first coil is now going to induce electricity in this coil. This electricity is going to be routed through a rectifier. Rectifier does the exact opposite of inverter. Inverter converts DC to AC, rectifier converts AC to DC, right? And that DC, and this can now be used for mobile phone charging, right? So this round coil is what you find at the base, at the back of the phones which have wireless charging facility, right? So many phones do have this wireless charging facility, and that is what is indicated by this round. Basically there is an induction plate which is kept underneath. Okay, that induction plate is nothing but a coil, and when you bring a similar magnetic field induced by an electric coil close to this induction plate, automatically that induces an electric current inside this, right? And that electric current goes through a rectifier which is kept inside the phone, and that charges the battery, right? So this is how wireless charging works. Wireless charging works on the same principle of Faraday's law. Okay, so that's the same principle that is used for magnetic stripe card readers as well. All right, so I hope you're clear. We'll move on to the next topic now. So this would be the last topic of discussion for the day. And why did I choose this particular topic? It's because this was a question that I always had, for which for some reason or the other I did not care to find an answer, or whenever I tried finding an answer, the research methods were not very systematic and I just dropped midway because I couldn't understand a lot of biology related terms, but now I made an attempt to try and understand what exactly it is. So I thought I'll share with you, and finding an answer for this question as to why and how antibiotics kill bacteria will also give you an idea about cell structure, right? And I believe cell structure knowledge was what could have helped you answer a 2020 question on Science and Tech, correct? Look at this question, it is totally technical. It's completely into biology, right? 3 statements are given, 3 statements are completely about cell structure. Just that the comparison that is made is between animal cell and plant cell. Plant cell has cellulose wall, whereas animal cells do not. Plant cells do not have plasma membrane, whereas animal cells do, right? So this is a completely a question that you could have handled only if you clearly knew what an animal cell is, plant cell is, right? So these kind of questions are what actually gives us this hint that UPSC expects us to know about the depth in areas that are very common to us. Of course, we all have been reading about cells, animal cell, plant cell, since our 6th standard onwards, right? So let's try to get a macro picture about this particular topic, okay? And in the process, we'll also be learning something about cells, right? So I've always wondered, when chemotherapy is given to cancer patients, the chemotherapy is simply what? You inject chemicals into your body, right? And those chemicals, they go and destroy the cancer cells. At the same time, they also destroy normal cells, healthy cells as well, correct? So that is the impact. So I've always thought that maybe antibiotics should also work in this similar way. Antibiotics, when we take antibiotics, let's say you have ear infection or you have throat infection, it's caused by bacteria. So we take a lot of antibiotics. There are broad spectrum antibiotics, there are specific antibiotics, right? We take azithromycin for sore throat. So all those antibiotics, they go and they clean the bacteria, they kill the bacteria, right? So there was always this concern as to why would these antibiotics, and how do these antibiotics reach the bacteria in the first place? They reach through the bloodstream. You consume an antibiotic, it gets dissolved in your abdomen, in your stomach, it reaches into the bloodstream, and from the bloodstream it reaches everywhere, every part of the body, and it goes and targets the bacteria, kills those bacteria, right? So if that is the case, then why would the same blood not take these antibiotics inside human cells, and why would human cells not be affected as a result of these antibiotics? That was the big question that I had, right? So for that, we have to understand the mechanism of action of these antibiotics, okay? So these antibiotics that you consume, it actually works, it kills bacteria in 3 different processes, right? One is by attacking the cell wall, other is by attacking the ribosomes, third is by attacking the DNA. Now you need to know what is a cell wall, what is a ribosome, what is a DNA, right? So we all know that cells are the fundamental units of every living organism, just like how atoms are the fundamental units of every matter. Similarly, cells are the fundamental units, and cells are all born, they are created, different cell specialization, it's all done when you are in your mother's womb, right? At that time itself, the cell specialization would have all happened. Certain cells become brain cells, certain cells become bone cells, certain cells become pancreatic cells, right? So each cell differentiation would have already happened, right? So when you are born, there are just so many cells. You are a package of so many number of cells, right? You are as a human being, we are multicellular organisms, correct? But that is not the case with bacteria. Bacteria is a unicellular organism. It's a single cellular organism, just one cell, not more than that. And just because we are multicellular, we have legs, we can locomote, we can move from one place to the other. But a bacteria is a single cell organism, so it cannot move. That's why nature has created with a tail, the flagellum, right? With the flagellum with which it actually moves. So human cells don't have a flagella, whereas bacteria has a flagella, which helps in its locomotion, right? It's a single cell structure, and bacteria were formed even long before human beings were born in this world, correct? So bacteria was the primitive life form. Human beings, we are more evolved life form, later life form. Unicellular organism, that's why it's called as prokaryotic. Human beings, we are multicellular, so we are called as eukaryotic life forms, right? So this is a eukaryotic cell, this is a prokaryotic cell. This is basically a bacterial cell, that's a human cell or an animal cell, okay? So the best way to visualize these two cells is, you can visualize this bacterial cell can be visualized as, let's say, something like, it's like a puri, okay, and a human cell can be visualized something like a gulab jamun. Now I hope you can understand the nature of these two cells. Okay, so for puri you have a covering at the top. The minute you press or you put a hole in that, the puri just collapses. Everything, all steam, everything just comes out of the puri, right? Whereas that's not the case with gulab jamun. You take a pin and you prick a gulab jamun, nothing is going to happen. The structure is going to remain intact. Why? Because it's a very, very solid mass, right? So that's the same case with the human cell. Okay, whereas a bacterial cell is not that. It's a very simple life form. So bacterial cell has an outer covering, what is called as a cell wall, right? And human cell, like a gulab jamun, doesn't have one specific covering as such. Okay, maybe the outer layer alone would have been brownish in color, but it's all the same. It's a one solid dense mass. Correct. So human cell doesn't actually have a cell wall. Same is the case with the animal cells. We don't have any cell walls, but it's a dense mass. This entire mass is what is called as endoplasmic reticulum. So basically you understand this is a solid mass which is called as endoplasmic reticulum. Okay, and the outer layer of that, just like a gulab jamun, it's the same flour, right? The outer layer has become burnt. Like that, the outer layer can be called as the plasma membrane. Okay, to this extent, let's just get to know about it. We don't have to, just for the sake of memorizing each and every component of human cell or animal cell, let's not get into that work. That is not the objective of this session. The objective of this session is to direct your thoughts starting from one topic as to how antibiotics are able to work and kill bacteria. For that purpose, we need to know this idea called as cell wall. Since we started discussing about the cell wall which is present in bacterial cell, we have to make a understanding that human cells do not have cell wall, because antibiotics work in such a way that it prevents the formation of cell wall, right? Bacteria tries to form new cell wall to protect itself. Those cell walls are made out of complex sugars basically, right? So bacteria tries to form new cell wall, but antibiotics, what does it do? It punctures this cell wall. It prevents the formation of cell wall. As a result of it, if the cell wall is prevented from forming, now what happens? This entire substance which is there inside it, it just collapses outside. That's how bacteria dies. Okay, that is one way of how antibiotics work and have their impact on bacteria, by preventing the formation of cell walls, right? And not only that, there are multiple ways how antibiotics work. There are different class of antibiotics. There are antibiotics called as beta lactams, which are like the first antibiotics to be discovered. Penicillin, which was discovered, was called as beta lactams. Its main work, how it achieves its work, is by destroying the cell wall formation. Okay, then there are other kinds of antibiotics which work in a different way, which work by affecting the ribosomes. Now let's try to understand what these ribosomes are. Ribosomes are common between human cell, animal cell, as well as in bacterial cell. Both these cells have ribosomes, right? What are these ribosomes? These okay, you see, inside a cell, in human cell, we have a nucleus, right? This nucleus, just like how in an atom you have a nucleus inside which you have proton and neutron, similarly the human cell nucleus, inside that you have this strand, right? The strand is what is called as DNA. Okay, all animal cells as well as bacteria, they have a strand of DNA. Even bacteria has a DNA. DNA, it's like the instruction that is given to every cell as to what to do in its lifetime. It's like the source code that nature has infused into each and every cell. It's like the big message that the creator has given to every cell as to what it should be doing, right? So the animal cell has DNA, bacterial cell also has DNA, just that animal cell has its DNA inside the nucleus, whereas bacteria has its DNA in that space. That space is what is called as cytoplasm, right? For animal cell we call that whole space is what is called as endoplasmic reticulum, right? Whereas for bacterial cell we call that empty space in which every other cell components are located. That space is what is called as cytoplasm, right? It is in the cytoplasm you have the DNA of the bacteria, whereas for human cells, animal cells, DNA is located inside the nucleus. All right, now what is the function of this DNA? This DNA has the biggest instructions given to it, right? Now this DNA, what it's going to do is, this DNA, it is going to send a message to ribosomes. Okay, in this context let's understand the function of ribosomes. Ribosomes, they manufacture proteins, right? Both in human cell as well as in animal cell. I'll give you an example for that. We cannot live without proteins. Today, if blood carries oxygen, it is because of hemoglobin. Hemoglobin is actually a protein. Okay, so what is a protein? A protein is basically, it's more like a band, it's more like a ribbon, right? And this protein, it has small collection of what is called as amino acids, right? These amino acids are basically organic substances, organic molecules, which are basically carbon, nitrogen, hydrogen combinations, right? So these amino acids are all located and they're arranged in a band, like in a ribbon like formation. This ribbon is what is called as a protein, right? So these proteins are very vital for our bodily functions. Without hemoglobin there is no transportation of oxygen, right? Insulin, which is produced in pancreatic cells. So hemoglobin is a protein which is produced in bone marrow cells. Okay, the bone marrow that is there inside the bones, right? So inside the bones you have bone marrow cells, and those cells have ribosomes. Those ribosomes will manufacture hemoglobin. Similarly, pancreatic cells in our pancreas will have ribosomes. Those ribosomes will manufacture insulin, right? So like that, every part of a body will manufacture very specific protein. Digestive system will digestive cells will manufacture the digestive enzymes. Okay, your bones, if they are firm, it is all because of the proteins that are manufactured by the bone cells, correct? So proteins are very vital for the functioning of our body. And where are the proteins manufactured? They are manufactured in the ribosomes. Okay, and how do ribosomes manufacture proteins? It's because of instructions sent from DNA. And how are instructions sent from DNA? We have what is called as mRNA, messenger ribonucleic acid. Okay, messenger. Okay, messenger means one who carries a message, right? That's what is called as mRNA. Okay, so the process through which DNA sends a message through mRNA to ribosome is what is called as transcription. Okay, the way how DNA sends a message through mRNA to the ribosome, this process is called as transcription. Okay, and when ribosomes manufacture proteins, it's called as translation, right? So I hope you're clear. The idea here is cells are the fundamental units of living beings, right? Animal cell, plant cell, bacterial unicell, all of these are made up of certain components inside, right? And all these components are located in different base layer. For human cells, animal cells, the base layer is called endoplasmic reticulum, and covering the cell we don't have any cell wall like how bacterial cell has. Even plant cells also have cell walls, Bacteria has cell walls. Human cells don't have cell walls. They only have plasma membrane. Okay, and it is inside the bacterial cell wall you have all the different components of bacteria located. Okay, so antibiotics, they destroy bacteria. For example, let's say you have the throat infection bacteria which is actually stuck to your throat. Okay, I think it's streptococci, I'm not very sure. So let's say the throat infection bacteria, it latches itself to your throat membranes, mucous membranes, and it's causing you strep throat. Okay, so now when you take antibiotics, azithromycin, it goes into the bloodstream, it goes into the throat membranes, and there it prevents the cell wall formation of bacteria, as a result of which the bacteria starts dying. Okay, another way of how some other antibacterial might work is basically by preventing the ribosomes from forming its essential proteins. Right, I told you hemoglobin, insulin, these are proteins of human body which are manufactured by the respective ribosomes of the respective cells. Similarly, bacterial cells ribosome would also manufacture the proteins necessary for the bacteria's survival. Now these antibiotics will go and block the mRNA reaching the ribosomes. So as a result of which ribosomes cannot manufacture protein, slowly bacterial cell will start dying. And not only that, another way of how antibiotics act, it's by going and destroying this RNA itself. Right, because I told you RNA is like the source code. Destroying the DNA itself. I told you DNA is the source code. Right, it's from here where every message goes to different parts of the cell. So if that memory itself is erased, the cell cannot function anymore. Right, that is another work that is done by these antibiotics. So how do they do that? They go and destroy the DNA, like how cancer cells. What are cancer cells? Cancer cells are basically mutations of your DNA. Right, when mutations happen to your DNA, your cell starts going crazy. It starts functioning in a very, very uncontrollable manner. Right, and deadly cells, cancer cells, they just start multiplying and finally they destroy the cell. Correct, that is exactly how the antibiotics work. They create mutation in the genetic component of the DNA. Okay, as a result of which the DNA gets repaired. Okay, sorry, it does not get repaired, as a result of which the DNA gets affected, and finally the DNA is not able to perform its function of sending message to ribosomes. Right, so it's by these three methods, as a result of which the antibiotics act on bacteria. Right, so this is how bacteria gets cleansed from our body. Right, so in this discussion we are able to understand how antibiotics function. In addition to that, we also have some sense of understanding of a human cell. We know that it does not have cell wall, it has a plasma membrane. It has a nucleus inside which DNA is located, whereas when it comes to bacteria, DNA is located in the cytoplasm. There is no such thing called as a nucleus over there. Both the cells have ribosomes, and ribosomes manufacture proteins. Proteins are very essential for various bodily functions. And how do ribosomes manufacture proteins? It's because of the instructions coming from DNA. And what carries this instruction? That is what is called as messenger RNA. Right, and this process wherein DNA sends message through making a copy of itself as a mRNA, as a messenger ribonucleic acid, it's what is called as transcription. Right, when the message is received by the ribosomes and when it starts making proteins, it's called as translation. Right, so these are some of the aspects about cell that we have covered as of now. Right, as a part of how we have covered the mechanism through which antibiotics act on bacteria, because that's also daily science. Right, we consume lot of antibiotics, and in the course of action, when the same antibiotics have been used in an improper way over a long period of time, automatically the bacteria itself becomes powerful, it becomes resistant to the antibiotic. Now antibiotic cannot break the cell wall, as a result of which bacteria continues to live. That is what is called antimicrobial resistance, which is actually emerging in various hospitals as a result of excessive use of antibiotics, as a result of self-medication. When you go to buy over-the-counter antibiotics, you self-medicate yourself, improper dosage. All these will lead to development of resistance of the bacteria. So that is another problem that's actually emerging in the recent years. Right. So I hope the session was useful to you in getting a glimpse of all the important science behind some daily aspects that we see in our life. If you would like to have a coverage of some more areas, so these are the ideas that I was able to think as to when I thought about what science and tech is there happening around me in my daily life. I could think of these, right? If you have some more areas to cover, maybe you can mention that in the comment section, right? And I'll try to cover it in the upcoming days, right? Thank you.